Hello everyone, welcome back to my channel X Gene and Protein X. In this tutorial video, I'm going to talk about monoclonal and polyclonal antibody and difference between monoclonal and polyclonal antibody, and additionally I'll talk about hybridoma technology precisely. So let's begin. So first we need to know that what is polyclonal antibody. So polyclonal, that name suggests that this kind of antibodies that produce from the multiple clones, and basically polyclonal antibodies are produced by injecting a specific antigen into animals such as rabbits and goats, etc. So initially these kind of animals are immunized with specific antigen, and this immunization process it occurs repeatedly to obtain higher titer of the antibodies. So after immunization process, 15 days later need to collect the blood, and then polyclonal antibodies are harvested from the anti-sera of the blood. So this is very simple process, and this is why polyclonal antibody can be produced very short period of time. So the harvested polyclonal antibody, these are produced from the multiple B cells clones. So these multiple clones, and each clones produce antibody against specific epitope. So this kind of polyclonal antibody, these are produced from the multiple clones. This is why it is known as the polyclonal antibody. And polyclonal antibody have better specificity over monoclonal antibody because they are produced by a large number of the B cell clones, each generating antibodies to a specific epitope. So there is not any single epitope. So there are different clones, it producing antibody in against different epitope. So next is the monoclonal antibody. So monoclonal antibody actually produced by the technology of hybridoma. So this is the monoclonal antibody very much specific that can be produced by the hybridoma technology. But polyclonal antibody, it does not require the hybridoma technology. So here I will just talk very precisely that what is hybridoma technology. So here initially the animals that are immunized with specific antigen, and then after 15 days spleens are collected from the animals. So because the B cells that produced and developed in the spleen, so this is why spleen are collected, and then B cells are isolated from the spleen. But these B cells, it cannot be survived in the cell culture because it cannot survive for longer time period. So this is why B cells fused with the myeloma cells. So basically the myeloma cells are the cancer cell. So this cancer cell, we know that cancer cell, it can grow very rapidly. So this is why the myeloma cells fuse together with the B cells, and then it form hybridoma cells. So you can understand that B cells and myeloma cells, it hybrid together. So this is why it are known as the hybridoma cells. And then the next process is the screening process. So by the ELISA technique, the higher antibody producing clones are need to select, and then that specific kind of clones need to grow in bulk amount to produce the monoclonal antibody, and that hybridoma cells need to be stored, and later on these hybridoma cells need to inject to the mice, and after 15 days need to collect the ascites from the peritoneal cavity. So these ascites contain lot of antibodies actually, and then this ascites it purified by the protein G bits or affinity chromatography. So this is a purification process. By this purification process, monoclonal antibody can be purified. So this is the entire hybridoma technology I just talked very precisely. So actually these are monoclonal antibody, these are the long process. So here this selection process, it is very important, and that the higher antibody producing cells or producing clones need to select by the ELISA, and then that single clone need to produce in higher amount to produce the monoclonal antibody. So these single clones that only can produce the antibody against single epitope. So this is why these antibodies are known as the monoclonal antibody, because this is produced from the only one single clone. So next is the difference in between monoclonal and polyclonal antibody. So in monoclonal antibody that made from the single clone, but the polyclonal antibody that made from the multiple clones, and it produced through the multiple steps of hybridoma technology, and this is why it needs longer process and it is very much expensive. But polyclonal antibody, this is very shorter process and easy to produce. It does not need hybridoma technology, and this is why it is not expensive. Sorry, here I mistakenly written here the expensive, but this is not expensive. And the next is the high specificity to a single epitope, because monoclonal antibody, this is produced from the only single clones. This is why the specificity to a single epitope. But in case of the polyclonal antibody, there is a higher antibody affinity against the antigen due to the recognition of multiple epitopes. And next is the, in case of the monoclonal antibody, there is the only single clones epitope. This is why the low cross reactivity and very low background noise. So in my later slide, I will discuss what is the background noise. I will show you the Western blot that picture. And in case of the polyclonal antibody, there is a high cross reactivity and very high background noise, because there are a lot of different clones that against different epitope. So this is why higher background noise is very much common in case of the polyclonal antibody. So here I will show you that there are two different images, and in the first image, that is the polyclonal antibody incubated membrane, and the second one is the monoclonal antibody incubated membrane. So you can see the difference here. In case of the polyclonal antibody incubated membrane, there are a lot of noise. You see, this membrane are not clear. So not only the band, so the beside the bands also there are a lot of bands, lot of noise. But in case of the monoclonal antibody incubated membrane, you can see these bands are very much clear and the membrane is also clear as well. So this is the main difference in case of the polyclonal and monoclonal antibody incubation. So this is very much important to select the right antibody for your Western blot or any other kind of experiment where you need to select the antigen or antibody. So here you need to determine that which kind of experiment you are doing. So if you do the Western blot, so I suggest always like monoclonal antibody, because you can get very much clear band. So in this case, so this is all about the polyclonal and monoclonal antibody. I hope this video will be helpful. So if you like this video, kindly hit the like button, share it, and please subscribe my channel. And if you have any comments, write in the comment sections. Thanks for watching.